flows from the afferent arteriole into a capillary bed located in the glomerular capsule. In the capillary bed, fenestrations, or small slits, in the capillaries allow small particles through. However, large particles, such as red blood cells, do not go through these fenestrations. The high pressure of the glomerulus assists in taking solutes out of the blood. Meanwhile, negatively charged glycoproteins in the basement membrane of the glomerulus prevent plasma proteins from being filtered out. Meanwhile, blood leaves the capillary bed and now travels to the efferent arteriole. This blood will provide necessary nutrients to the cells surrounding the nephron. The resulting mixture of water and solutes, or the filtrate, now travels to the proximal convoluted tubule. The wall of the proximal convoluted tubule is a single cell thick. The filtrate flows through the lumen of the ring of cells, carrying water, salt ions, and glucose. The inner portion of the cell contains microvilli that increase the surface area for reabsorption. Several transport mechanisms are used in order to accomplish reabsorption of salt ions, water, and glucose. The majority of salt ions, being sodium ions, chlorine ions, and potassium ions, leave the filtrate and return to the bloodstream. The mitochondria in the cells provide energy for active transport of the salt ions into the intercellular fluid outside the tubule, which is then taken by the peritubular capillary bed because water moves from a hypotonic region to a hypertonic region. Water leaves the filtrate in the same route as the solute by osmosis. Finally, all glucose that is in the glomerular filtrate is absorbed into the bloodstream through active transport. Water and remaining dissolved solute enters the descending loop of Henle. The loop of Henle has three parts: descending loop, ascending loop, and loop. The filtrate travels down the descending loop of Henle. The descending loop of Henle is permeable to water, but not to ions. Therefore, as the filtrate travels, the water leaves the descending loop of Henle by osmosis. The filtrate travels from the descending loop to the ascending loop through the loop. The filtrate travels up the ascending loop of Henle. This part of the loop is permeable to salt ions but impermeable to water. Ions thus leave the loop into the intercellular fluid of the kidney, making that intercellular fluid relatively hypertonic. The filtrate then travels to the distal convoluted tubule, or DCT. The distal convoluted tubule reabsorbs sodium ions through the coupled secretion of hydrogen ions and potassium ions. This plays an important role in affecting the acidity of urine. The filtrate then enters the collecting duct after leaving the DCT. From the hypotonic filtrate that enters from the distal convoluted tubule into a nearby collecting duct, some water is reabsorbed. The collecting duct is differentially permeable to water. The permeability of the collecting duct is linked to antidiuretic hormone, or ADH. ADH is released from the pituitary gland in the brain. The permeability of the collecting duct is linked to antidiuretic hormone, or ADH. ADH is released from the pituitary gland in the brain. If ADH is present, this hormone makes the duct permeable to water. As a result, water exits out of the duct by osmosis and into the peritubular capillary bed and bloodstream. If ADH is not present, the collecting duct is impermeable to water. Water that stays in the duct is passed as urine.